everyone, I am Professor Charu Dogra from Ramjas College, University of Delhi, and continuing in the series of biotechnology, I am discussing about the molecular diagnosis method for diagnosing various genetic diseases. Now, these molecular biology tools and techniques that have been developed help in the diagnosis, the prenatal diagnosis of various genetic diseases such as the cystic fibrosis, sickle cell anemia or beta thalassemia. So, I had already discussed in my previous sessions about the RFLP method and the SSCP method, the restriction fragment length polymorphism based method or single strand conformation polymorphism based methods. In this session, I will be discussing about another method which is termed as the ASO dot blot method. Now, what does this ASO stands for? ASO is allele specific oligonucleotide, and this technique utilizes the synthetic oligonucleotide probes which are about 19 residues, one which is complementary to the normal and one to the mutant allele. So, these synthetic oligonucleotide probes are synthesized by DNA synthesis procedures, and these probes are complementary to the gene of interest that we want to study. So, in a particular disease, the gene that is responsible, for example, in cystic fibrosis, the CFTR gene, or in sickle cell anemia, the hemoglobin gene which we are talking about, or in beta thalassemia, the beta thalassemia gene that we talk about, then the probes are generated or artificially synthesized for these particular genes and one for the complementary to the normal genes. Now, under appropriate conditions of hybridization, the oligonucleotides will bind only when there is complete complementarity. So, this is a very simple schematic representation which tells that this is a wild type gene and this is a wild type probe, and therefore, because of the complementarity it binds to it. But if there is a mutation, for example, in this case there is a mutation generally in the center, if there is a mutation present in a particular gene, then this normal probe will not be able to bind, and if a mutant probe is also generated, then it is going to bind to this one. And obviously the normal and mutant can be tagged with different molecules or different signaling molecules such as the fluorescence or radioactivity or enzyme substrate based complexes, and then they can be analyzed to detect whether the gene is a normal gene or whether the gene is a mutated gene, and therefore the genetic disease can be diagnosed. So, this is the ASO thing. Now, looking at the dot blot thing, that what happens in the dot blot is when the DNA is extracted and they are blotted on, for example, the nylon filters, and then the denaturation is done by alkali. So, it is again a blot method, a blotting method. We have already discussed about the southern blotting, the northern blotting method, or the even western blotting method for the identification of various sequences in the DNA, RNA or certain protein molecules. For that, what essential step was the blotting of these DNA or transferring of these DNA onto the filter papers, or basically nylon membranes, or blotting it onto that. So, in this case also we are blotting the genomic DNA or the amplicons on the nylon fiber, but it is said to be dot blot because they are not run on the agarose gel, but they are basically loaded onto as dots. So, these are the nylon fibers in which the DNA extracted from different samples is transferred to in the form of dots, and as it is processed in various hybridizations, nucleic acid hybridization, because we are talking about DNA here. The denaturation is done by alkali, and then the hybridization is done with tagged probes. It can be radioactive, it can be fluorescent, it can be again that substrate enzyme thing, the chemical tagged probe, and then it is identified, and then the washing up is done so that the non-specific binding is removed, and then it is developed. So, if it is radioactive, then it is developed by in a certain manner. If it is fluorescence, then the fluorescence label is detected in the scanning machines, or if it is a chemical method, then enzyme substrate complex chemical reaction is done so that it gives you the certain signal. So, this is the autoradiography of the blots which is done. So, this is the dot blot method. The blotting is the same, the transfer, and the processing is also the same, just like different nucleic acid hybridizations. The thing is that the entire DNA or whatever it is transferred as dots onto the membrane. So, this is the dot blot method. Now, I will again look at the two different diseases, which is the sickle cell anemia or cystic fibrosis, which for the diagnosis using this ASO dot blot method. Now, just to review, the sickle cell anemia is basically because of the difference or the mutation in the 6th codon in the hemoglobin gene, and because of that the amino acid is changed, and this leads to the polymerization upon oxidation of the hemoglobin S molecules, and leading to this kind of a sickle shaped RBCs rather than the normal RBCs. So, these are the 4 chains of a normal hemoglobin, and this is the hemoglobin chains for the sickle cell anemic patient, and upon oxidation it is polymerizing, and therefore the shape is changing. Not only the shape, but the capacity to carry the oxygen also changes, and therefore it leads to various clinical manifestation for the sickle cell anemia. So, this is the general basis of the sickle cell anemia. Now, let us look at how the allele specific oligonucleotide dot blot method is used to diagnose this particular disease. Now, this is the dot blot analysis of amplified genomic DNA. Now, the A1 is probed by a normal ASO probe, and the B1 is probed by a mutant probe. Now, please understand this very carefully, that in the figure A, there are 2 blots which are given. One is the lane 1 and other is 2. Lane 1 is a normal clone and done at different dilutions. So, different dilutions means that the amount of DNA, the PCR amplicon that has been loaded in these 3 dots is in the increasing concentration from lower to the upper thing. So, this is lesser amount of amplicon, this is more, and this is the largest amount of volume of amplicon that has been loaded. The same in this case, and again in the same way here. But the thing is that this is a normal clone or colony or sample that we are using, and this is the DNA amplicon from the mutant colony, and this can be, if in the prenatal diagnosis, this can be the normal parent or whatever, and or a normal individual, and this can be the fetal DNA that we can do, and therefore identify whether it contains a mutation or not. Now, the ASO probe is used. Now, this again I repeat that ASO is allele specific oligonucleotide. So, this is a normal ASO probe that we are using. So, normal ASO probe against this normal DNA sequence which is there. So, this probe will be binding to this particular DNA sequence, and the complementarity, again I remind you that how you can alter the complementary binding of the probe to the DNA sequence is by changing the stringency of binding. So, remember the word stringency, that based on certain conditions the stringency can be increased or decreased. Stringency refers to the binding of the probe to the nucleotide sequence, whether you require the 100% complementarity or you are lenient enough so that 2 or 3 mismatches can be accommodated and still the binding will occur. So, in this case we do not allow any stringency, do not allow any leniency, and it is a highly stringent conditions that we give for the binding of the probe to the desired nucleotide sequence in the 100% complementarity matter, because there is only a single nucleotide polymorphism or single nucleotide change that is occurring here. So, it does not allow any deviation from the sequence of the probe and the sequence of the DNA sequence or the gene that we are targeting at. So, this is the normal sequence and this is basically the mutated sequence. So, a normal ASO probe will be this particular ASO artificially synthesized nucleotide sequence which is complementary to the normal gene. And a mutant ASO probe will be again an artificially synthesized nucleotide sequence which will carry this mutation that is present here. So, I hope you understand what the normal ASO probe or the mutant ASO probe would mean. Now, normal ASO probe under 100% stringent condition is going to bind to the normal sequence, but it is not going to bind to the mutated sequence. And a mutant ASO probe under again 100% stringency complementarity conditions is going to bind to the mutant sequence, but not bind to the normal sequence. So, this is the conceptual understanding behind it. So, if you look at these two dot plots, what is happening here is when you are using a normal probe, it is showing the binding. You can see the signal is present here. Some background signal is also present here at higher concentrations, but certain signal is present in the normal allele, because the normal ASO probe is going to bind to the normal amplicon, but not to the mutated amplicon. And in this case you are using a mutant ASO probe. So, the mutant ASO probe will not bind to the normal clone, but is going to bind to the mutant alone. So, obviously there is a difference in the signaling and difference in the detection where the probe is binding will lead us to diagnose that the DNA or the amplicon is from a normal individual or is carrying a mutation and may have the probability or may have the particular or is inheriting the genetic disorder that we are talking about. So, in this case it is a sickle cell anemia example that I have given it here. Then there is again the cystic fibrosis diagnosis. As I said, we are looking at the diagnosis of these two important genetic disorders and understanding the application of the different techniques that have been developed. So, this is the cystic fibrosis diagnosis. Again, just to brush up a little of your memory, that the cystic fibrosis arises because of the various mutations. Now, these mutations in the sequences, it can be the missense mutation or it can be a nonsense mutation where it leads to the occurrence of a stop codon, or a missense mutation where one amino acid is displaced by another amino acid. There are 6 kinds of mutations. They are classified into 6 categories based upon the functional differences that it is going to give rise to. So, we are talking about the CFTR gene here. This is the normal CFTR gene, which is a transmembrane protein regulator. If you remember, it allows the transfer of the chloride ions across the cell. So, this is a normal cystic fibrosis transmembrane regulator protein, and this is the normal one. But if there are certain mutations, then there is the absence of this particular protein, or there is no trafficking of this protein to the transmembrane. You remember, this is the main and most prevalent mutation leading to the cystic fibrosis, the delta F508 mutation, and this mutation leads to non-trafficking of the protein to the transmembrane. So, the protein is formed, CFTR protein is formed, but it is not transported to the transmembrane and therefore cannot cause the conduction of the chloride ions. Then there is also that there is no function. So, these mutations leads to no functioning of the CFTR protein. So, the protein is formed, it is transported to the transmembrane, but it is not functioning properly. Then these mutations leads to less functioning of the protein molecule. So, as you can see that the CFTR proteins are there, but because of the alteration of certain amino acids, the conduction is not as such which is present in the normal individuals. So, this is again a problem. Then there can be a low protein that is present. So, the amount of protein is low here, or there can be again the degradation of the protein that has occurred. It is present there, but then it is processed in the degradative pathway and the CFTR gene is degraded. So, all these leads to the genetic disorder cystic fibrosis with various varying clinical manifestations and based upon the different kinds of mutations. So, these are the mutations, these are just given examples, but there are many genetic variants or mutations of cystic fibrosis. There is a whole spectrum of mutations that can cause to the disease cystic fibrosis. Now, let us look at the diagnosis of cystic fibrosis by the ASO dot blot method. So, this is the ASO dot blot of cystic fibrosis mutations. Now, this is again a very important dot blot and I am going to very slowly explain what is happening here. The two things that you have to remember here is that some of the blots will be of the normal individuals and some of the blots will be for the cystic fibrosis mutations. Okay? So, these A, B, C, D, E are the different mutations that we are talking about. So, I am not going into the research aspect details of all these mutations. We are not dealing with that because we are just trying to understand the technique and the concept behind the technique. So, just consider this A, B, C, D, E as the different mutations that we have talked about here. So, they are different mutations that are basically the A, B, C, D, E, and we are going to probe it with these different mutant probes that we are going to use. So, again these are different mutations and we are looking at the dot blot of these mutations. So, that was the one aspect. And the second aspect that you need to keep in mind to understand this blot is the inheritance pattern. So, I again repeat, it is a recessive genetic disorder. If the two parents are carrier parents, then they are going to give rise to the offsprings where there can be a possibility of a homozygous dominant individual, there can be a possibility of an heterozygous individual, and there can be a possibility of the homozygous recessive individual. Now, again remember that in homozygous dominant, both the alleles will be normal. In heterozygous individuals, one allele will be normal and therefore hybridized to the normal probe, and one allele will be mutant and will therefore hybridize with the mutant probe. And in the third one, the homozygous recessive condition, there will be no normal sequence that would be present and both the alleles will be the mutant alleles. So, there will be no signal in the normal probe, but there will be a signal in the ASO probe, mutant probe that we will be using. So, remember these 2 points, and now let us analyze this dot blot which is presented in this particular slide. Now, there are 4 samples that we are analyzing. So, A, B, C, D, E are the different mutations, and these different mutations are different probes are used to ascertain these mutations, and 1, 2, 3, 4 are basically the samples that we are analyzing. So, when we are analyzing the samples, now there are 2 columns, there are 2 lanes which are shown here. So, the first lane is probed with the ASO normal probe for this particular gene, and this is probed by the mutant probe. So, I hope again this is very clear to you. So, this is the normal ASO probe, this is the mutant ASO probe. The mutant ASO probe, the upper one is for the mutation A, the mutant probe for B, the mutation C, the mutation D and the mutation E. So, these are the ones which are there. So, this is one allele, this is the other allele that we are talking about. Now, look at the lane number 1 for the mutation A that we are talking about. Now, this lane number 1 is going a signal here as well as is giving a signal here. So, it is giving a signal with the normal ASO probe, and it is also giving a signal with a mutant ASO probe for the presence of this mutation in the transferase gene. So, what do you understand by that? That individual 1 is heterozygous for the particular mutation which is termed as A. I hope it is clear to you now, because it is giving a signal in the normal allele ASO probe, and it is also giving a signal in the mutant allele specific oligonucleotide probe. So, when a normal and when a mutant allele is present, then that is a heterozygote individual for this particular mutation. Now, look at the number 2. So, 2 is again heterozygous because it is giving signal in the normal ASO probe as well as it is giving signal for the mutant ASO probe. Now, look at what is 3. So, can you just identify this yourself? The 3, there is no signal in the normal probe, but there is a signal in the mutant ASO probe. So, what would it be? It would be a homozygous mutant, and therefore, it will be carrying the particular disease in particular to this mutation. Now, this mutant we can identify that what mutation we are talking about here. and therefore we can also diagnose the detail here, that what function is affected, so that when we look at the gene therapy or when we look at the different kind of functional makeup that we need to do for its proper functioning, we know which function has to be addressed. So, if it is this particular mutation, that we know that there is basically no functioning that is occurring, or if this is this particular mutation, we know. So, we identify this on the basis of this. So, now look at the B mutation. The number 1 and 2 are heterozygotes for the B mutation. Number 3 is homozygous for the B mutation, that it is carrying the disease, and number 4 is a normal individual. So, in both these cases, number 4 is the normal individual because it is not carrying the mutant allele, but it is carrying only the normal allele. So, number C mutation, the 1 and 2 are normal individuals because there is no signal in the 1 and 2 lane with the mutant probe. So, it contains only the normal sequence. Number 3, there is no signal here, there is signal present here, it is a homozygous mutant. Number 4, signal here, it is a heterozygote carrier for the particular cystic fibrosis genetic disorder. Look at the number D. So, in D again 1 and 2 are normal and 3 and 4 are heterozygotes. In number E, 1 and 2 are normal and 3 and 4 are heterozygotes for this particular mutation in the CFTR gene. So, this is a very useful and very depicting method, and I hope I could explain it well, that how ASO dot blot method could identify 5 CF mutations in a particular in various individuals. So, this is the method for this thing. So, the mutants were hybridized with the wild type oligonucleotides, as I said, the normal ASO probes, and with the mutated oligonucleotide probe. The numbers 1 to 4 refer to the single individual for each of the tested mutation. So, this is again a very, very important technique to identify this particular thing. Now, another just a deviation or an extension for this dot blot method is used, which is termed as the reverse dot plot method. So, what happened in the reverse dot blot method is that allele specific oligonucleotides are immobilized onto the nylon membrane and the patient specific PCR product is used as the probe. So, in here what is happening, that the patient specific PCR amplicons are loaded onto the are transferred to the nylon membrane and ASO probes are used to identify. But if we have chips where the ASO probe, a normal probe and a mutated probe was on the membrane, and then we use the patient specific. So, this is a reverse dot blot analysis which is used here. It allows several different mutations to be analyzed simultaneously. So, you do not require that in the each genomic DNA we use a mutant probe to identify. We can in fact have chips where different mutant probes are loaded onto the membrane and the patient DNA amplicon is loaded onto that and therefore identified where the signal is coming. So, it is termed as the reverse dot blot method. So, this is again a pictorial representation of the reverse dot blot analysis, where the upper lane is the normal lane. So, this is the normal probe and this is the mutated probe that is loaded, and in the normal probe and the mutated probe, the patient amplicon for that particular gene is transferred. So, when this is transferred, and this 1, 2, 3, 4, 5 is the different kind of mutations which are present in here. This is a normal ASO probe, but in mutated probe, this can be because of one mutation, another mutation, another mutation, another mutation, another mutation. So, various mutations are loaded. All you do is you isolate this DNA, and this DNA then you biotinylated you are using. Again I said this is for the detection method, it could carry the radioactive molecule, or it could carry the biotinylated for the chemical based detection, or it could carry the fluorescent probe for fluorescent detection. So, this is when transferred or probed, when this particular nylon membrane is probed with the PCR amplified single stranded, and this is also single stranded, then you can see such kind of patterns observed. So, I hope you can interpret this thing, that the signal is coming both in the normal and the mutated probe. So, this particular patient is heterozygote for this mutation number 1. It is heterozygote for mutation number 2. It is homozygote for a mutation number 3. So, this mutation number 3 is causing the CFTR in this particular individual, because there is no normal allele which is present here. This is a normal individual, no mutant probe is identifying here. This is again heterozygote for mutation number 5. So, several different mutations can be identified simultaneously, and simultaneously one DNA can be identified for various. In this case, it is carrier for these mutations, but the disease is manifested because of this particular mutation, which is mutation number 3. And as I said, this mutation number 3 can be anything, the missense mutation, or we know that because we are using that probe which has been generated against that mutation. So, this is a reverse dot blot analysis. So, in the last I discussed a real example for this dot blot method, the hybridization probes complementary to the 10 most common Thai beta thalassemia mutations. So, as you can see here, these are the beta thalassemia mutations that we are talking about. These are the 10 lanes, and in each strip I am taking these real examples, so that you do not learn and understand about the concept of the technique just theoretically. These are the real blots, these were the real experiment that was done, and these are the real blots that appear. So, you have an understanding of how actually in lab it comes, and you are able to analyze that as well. So, you are not just understanding the concept theoretically, but practically also you are going to do that. I would always suggest that you independently look at those blots and try to interpret the outcome based on your understanding what I have talked about today. So, looking at this picture, it is identifying these 10 markers. These are the 10 most common in Thai people, the beta thalassemic mutations which are there. And in each strip there are 2 lanes, one upper lane and one lower lane. So, in upper lane the normal DNA is plotted, and in the lower lane the mutated DNA. So, mutated DNA for this probe, the mutated DNA for this one, mutated DNA for this, mutated DNA for this, and so they have been done. And when you process that, then you recognize that in, for example, this topmost strip, the signal is present in the normal lane and there is no signal which is present in the mutated lane. So, in this particular, the patient DNA that you have used, no mutation is present. All these 10 mutations are not present, but it is a normal individual, because only the normal ASO probe bind to, or basically because the probe is present here, only the patient amplicon bound to, or the individual amplicon bind to only the normal DNA and not the mutated DNA. Look at the one below it. All the signal for all these mutations are coming in the normal topmost lane. But in the lower lane, you can see this particular signal here. So, this individual is mutated for this particular allele which is present here. So, you can obviously diagnose that it is carrying the beta thalassemic mutation, which is this mutation, this 28 mutation which is there. There are different things in here. Again, you can see that this mutation is present in this individual, while at all other alleles it is a normal individual. Similarly, in this, all the alleles are normal individuals, but this is the mutant allele. All the alleles are normal, but this mutant allele. In here, all the alleles of normal as well as the mutant allele for these two. So, what does it interpret? That this particular patient is heterozygote or carrier for these two mutations, because it is carrying one normal allele and one mutated allele. So, these dot blots, these are very, very important, and which is for the understanding. This is again rest of them. So, again you can see all the normal strips and a mutated strip, a mutated signal, all the normal signals. So, where there are the signals in the normal as well as in the mutated, that means it is heterozygote for that mutation. So, this is heterozygous for this mutation, heterozygous for this mutation, homozygous for this mutation, and when there is no mutant the DNA is not binding to any mutant ASO probe, then it is normal for all those alleles. The individual which is not carrying any mutation is a normal one, and it is carrying any one of the mutations which is leading to one of the dysfunction of the CFTR gene. It is going to manifest the the cystic fibrosis disease, and it is basically going to give rise to the, or we can diagnose the presence of that genetic disorder which is there. So, today I have discussed about two important molecular biology tool and technique that is used for the diagnosis of the genetic diseases. The first was the PCR SSCP, which is a single strand conformation polymorphism, and the second was the ASO dot blot method, where allele specific oligonucleotides was used in dot blot methodology to identify the occurrence of the diseases. And I took specific examples of cystic fibrosis and sickle cell anemia and beta thalassemia mutation to present the application of these techniques for the molecular diagnosis. We continue with this in our next sessions. Thank you.